Hello everyone. Hi. Yes. Hi. Am I audible? I hope I'm audible. Give me a thumbs up, please. If the audio is good, give me a thumbs up, please. Yeah. So we're good to go, right? Okay, so this is me, Alka Rajimon, your biology tutor from GoScore. We are the fastest growing edtech in GCC, and our aim is to provide quality education for all. Yes, here I am with another new chapter which we are gonna learn in 2-3 parts, just like how we covered up heredity. I hope you all remember the heredity portions still. Okay, can you just say hi? Yes, hello. Okay. Thank you so much for joining the live. Thank you. So yes, I hope you all have made good use of our previous chapter, heredity. I hope the questions were useful. I hope you all had some fun in between. Yes, so today I'm here with a new chapter. It's none other than one of the most important chapters that you have in your grade 10, which is chapter 7, how do organisms reproduce? Yes. So, are you all ready for the new session? If you all are ready, if you all are here, without wasting much time, let's begin the session. So we're going to discuss some of the most important concepts in the chapter. Along with that, we'll be discussing PYQs, which are the previous year questions. So this is filtered questions that we have chosen from the previous years, which have been repeatedly asked, frequently seen, and most recently asked questions in the boards. So please make use of the session. I hope you all will be staying with me along the session for next 1 hour. Yes. Okay. So in this chapter, the major content we have to discuss are blueprints, cell, chromosome, DNA. I hope you already know about the differences between cell, chromosome, DNA by the time, and variation, modes of asexual reproduction, sexual reproduction in plants, sexual reproduction in humans, and reproductive health. So if you simply look into the content, you can understand that there is basically this classification. So blueprint, cell, variation, it's all an intro into the chapter where you will get an idea regarding what DNA, chromosome, all those stuff are. And then you have asexual and sexual reproduction, which is the reproduction, the major content of the chapter. And then finally, there is an extra topic which is reproductive health, which is very less in content, but still, if you're lucky enough, you'll be getting 1 or 2 questions. Like, it depends. We have seen questions from that part, but it need not to be very frequently asked or anything. Okay, so this is a quick idea regarding the chapter. Let's move into the chapter. Since this is the chapter, how do organisms reproduce? Let's begin the class with the topic reproduction. So as we all know, reproduction is nothing but a biological process by which new individuals of offspring are produced. So reproduction is nothing but a biological process which leads to the production of next generation. So there are a lot of events happening in between, and there will be a lot of questions regarding those happenings. So let's see one question from that particular part. This is a question. Reproduction is one of the most important characteristics of living beings. Give 4 reasons in support of the statement. In support of the statement. Yes. Are you able to answer anything from that part? Anything from this part? So reproduction is an important characteristic of living beings, just like growing, breathing, digestion happening, metabolism happening. Reproduction is also a part of a living organism's characteristic. So they have asked for reasons, and they have actually asked specifically for 4 reasons. So if such a question comes, this is how you can answer it confidently. Make sure these points are included in one way or the other. So the first and most important point should be existence and continuity of a species. So as long as a particular animal or a plant keeps reproducing, it is never going to get extinct. That is, the species keep going one generation after the another, and it will be in a continuity. So continuity of species is the first and most important point regarding reproduction. Second thing, pass genetic information from one generation to next generation. So passing of genetic information, that is, whatever traits I have in me can be given to the next generation or the future generations of Homo sapiens only through sexual reproduction. That's the only method by which I can transfer the good traits that I might have in my DNA to the next generation. So, passing of the genetic information, let it be some mutation which is positive, let it be some variation that is going to help us to survive the future. So anything like that, any sort of such genetic information can be transferred from one generation to the next only through the method of reproduction, which is sexual reproduction. And then it brings variation in the next generation, which is the basic of evolution. So variation happens in sexual reproduction. So reproduction can be sexual or asexual, but variation would be possible only through sexual reproduction. So we'll see what variation is and how it is occurring in sexual reproduction and all in the upcoming classes. So for now, let's say happening of variation, or you can say differences. I think in the heredity chapter, we have already covered what variation is. So you all might already know what variation is. So yes, so variation is the third uh, point which you should be explaining for that question. And finally, if reproduction doesn't take place, no living being will survive on earth and the organism will go extinct. So saving a species from extinction is done only through reproduction. No matter you are a living being or not, whether you breathe or not, whether you survive or not, if you're not able to reproduce and make a next progenies, you are going to extinction. So saving any species from extinction, or that DNA itself, that gene pool itself getting extinct, is covered up only through a method of reproduction. So these are the points you can easily put out into the paper, into the answer paper. So make sure you put out those in that particular elaborated manner as per the question, like as per the mark they have asked. Okay, and this is another question. Newly formed DNA copies may not be identical at times and give reasons. So what is DNA copying? Suppose there is a cell. Suppose there is a cell. Cell has a nucleus. Inside the nucleus, there is definitely the thing called DNA, right? We have these chromosomes inside which the DNA can be seen. So when this particular cell is dividing, when this particular cell is dividing and making new copies of the cell, the DNA is also dividing and replicating and creating new copies of it. But the question is, newly formed DNA copies may not be identical. So how can that happen? So when the DNA copying happens in the new daughter cell, the DNA may not be copied, be exactly equal to the DNA which is present in the parent cell. So there can be differences in the DNA. So this red color which shows here, it is actually showing a variation or a difference. So the question is, how come this is happening? Okay, so let's see how the DNA copies are not identical at times. So the DNA copying mechanism may not be same for all cells. When a cell reproduces, when a cell splits and becomes two daughter cells, just like I just drew a figure there. When a cell reproduces, DNA replication occurs, which results in the formation of two similar copies of DNA. The process of copying the DNA leads to some variations each time. So what happens here is that, suppose this is the parental DNA, and when this DNA is splitting and becoming new copies of DNA in the new parent, what happens is there is a possibility of occurrence of a new mutation or maybe a new variation in this new piece of DNA. So that red spot here indicates a new variation which wasn't present in the parental DNA. So that can happen. It is possible to happen. So the process of copying DNA leads to some variation each time that it's getting replicated. No biochemical reaction is absolutely reliable. It can be different at different times. Therefore, it is only to be expected that the process of copying DNA will have some variation. So this is the key word, variation. That is the key word your examiner or the person who is evaluating your answer sheet is looking for. So if you're able to explain that single term there, when DNA is replicated, why it is not similar to parental DNA? Because variation has happened there. Maybe some mutation has happened. Maybe there has been a sexual recombination happened there. So that's what you have to explain there. As a result, the DNA copies generated will be similar but may not be identical. That's the second terminology. The DNA which is actually replicating and is producing a new copy, it can be similar to the parent. The new DNA babies can be similar to the parent, but it is not at all identical. It's not same. Differences can be there. I hope it's clear. Now, before going to the next question, let me just go through the basic concept of this chapter. When this chapter is explained by the NCRT in the beginning itself, they have actually introduced the term blueprint. So, since you guys are already going through your revision sessions, you might already know what a blueprint and all is. So, in biological term, in our case, blueprint of a living organism. Blueprint of a living organism is nothing but your DNA, because this is the same DNA that will be carrying all kinds of, yes, so this will be the DNA that will be carrying all the details of your body. It could be the traits you have, it could be the skin color, your hair color, maybe the enzymes produced inside your body, the HCl in your stomach, maybe the saliva in your mouth. It's all encoded inside a small chemical structure called deoxyribonucleic acid. And this deoxyribonucleic acid, which is almost a double helical structure, is situated inside a structure called chromosome. Inside this structure, you can see the double helical structure. So this chromosome is again kept inside a cell organelle called the chromosome. So as you can see here, these X-shaped chromosomes, this is a condensed form of chromosome. This is again kept inside a small globular 3D structure called nucleus, which is a controlling network of your cell inside your body. And this entire nucleus is kept inside your cell. So this is basically how the DNAs are arranged. Inside the cell, there is nucleus. Inside the nucleus, you have chromosome. Inside the chromosome, you have deoxyribonucleic acid. So each and every single part of this nucleic acid will be carrying some or other trait. So we call a portion of that DNA as gene, which will be encoding a trait. It could be a protein, it could be an enzyme, etc. So there would always be a gene product out of this DNA. So this is the basic concept of blueprint. So no matter what kind of question comes with the word blueprint, this is what you have to explain. Is that clear? I hope it's clear, and I'm moving on. Okay, so this is another question which is asked regarding the same content that we have just discussed. Okay, let me see my kids. Hi, hello. Yes, hello. Who is asking photosynthesis now? We are discussing such a great topic, reproduction, and you want photosynthesis now? Okay, we'll come to photosynthesis later, dear. Okay. So thank you all so much. Thank you all for joining. I mean, seeing you all in the comment section itself is a great energy. So thank you so much. Okay, when a cell reproduces, what happens to its DNA? It's the same kind of content, the same thing that we have just explained. The theory is same, but the question has come in a different way. It's just a one more question. A cell reproduces. What happens to DNA? You can expect maybe a 2 marks maximum to that question. That's it. So what happens to a cell when it reproduces? Nothing much happens. When a cell reproduces, the DNA inside it also replicates. It's called DNA replication. DNA multiplies, right? Okay, so when a cell reproduces, the DNA replication occurs, which forms two similar copies of DNA. And you can add on there, since you already know, these DNA copies may be similar, but not identical. But not identical. Yes, these DNA copies can be similar, but they are never identical, not same. Okay, fine. Now this is another question. Let's go through it. It looks so big. Let's read it. So what is the effect of DNA copying, which is not perfectly accurate on the reproduction process? So DNA copying, this DNA replication happening, is not a biochemical process which is perfectly accurate. Okay, that's the thing. How does the amount of DNA remain constant through each new generation, which is a combination of DNA copies of two individuals? So that's the question. We have to answer two things. What is the effect of DNA copying which is not perfectly accurate? So what happens when this parental DNA is replicating and creating two daughter DNAs inside the new daughter cells? The first question is, this is not a perfect process. So what happens since that process is not perfect? Which means since the newborn DNA material is not identical to the parent, what is the biological process happening there? That's the first part of the question. I think you already got the answer. And the second thing is, how does the amount of DNA remain constant through each generation, which is a combination of DNA copy of two individuals? Since suppose it's a sexual reproduction, there would definitely be a male gamete and there would be a female gamete. So how come both these gametes are contributing to a DNA content which is always constant? How does the DNA content always stay constant? The amount of DNA is always constant. It's not getting double or it's not getting halved or anything. So how is it happening? I think the question is clear. It looks like a big question. The answer is simple. Reproduction results in the production of offspring similar to parents' body design and form. The exact blueprints for body designs are inherited in the offspring due to DNA replication or DNA copying. So nothing, it's just an intro. This is just a framework of how you can explain that particular question. When you're answering by yourself, you can use your own words. So just in the first sentence is just explain DNA replication or DNA copying is a mechanism where the parent's DNA is just replicated to the offspring. That's it. No biochemical reaction is absolutely reliable. We already said it in the previous answer. The same thing. So this process is not perfectly accurate. As a result, the DNA copies generated will be similar, but they may not be identical. So again, the same thing. So the first part of the question, which is asking, what happens since the DNA copying is not perfectly accurate? What is the effect? The effect is that since the DNA copying is not perfect, the DNA produced would not be similar to the parental DNA. Sorry, it would be similar to the parental DNA, but not identical. That's it. So this imperfect DNA copying results in genetic variation. So that's the point you have to talk there. Again, the same theory, but the question is asked in a different way. You just have to identify the question, what they're asking about, right? So you just have to say one point, variation would happen since the DNA mechanism, copying mechanism is not perfect. Variation can happen and the DNA produced wouldn't be identical to the parental DNA, but it will be similar. That's just the 2 points you have to explain there. Then There is a second part of the question asking, what is it? What's the second part? How does the DNA remain constant through each new generation? How is that DNA content maintained through each and every generation? So, combination of DNA copies of two individuals, let it be male and female, occurs during sexual reproduction. Reduction division during gamete formation halves the chromosome number in both male and female gamete. So I think we already know the process called meiosis. We have already studied it. So meiosis is a kind of cell division which specifically happens only in your reproductive cells. Suppose in a man, it would be in the case of sperm formation, and in a female, in a woman, it would be happening in the time of ovum formation. So the sperm and the egg would have, so this sperm and egg would have only half of the chromosome, that is 23 chromosome. So only half of the DNA content would be there in both the gametes. And when they fuse together to form a zygote, this 23 plus 23 would create a 46 number of chromosome, that is the full content of DNA. And again, when the zygote is becoming a baby and that baby grows up into an adult and that adult is giving birth to another baby, again the same meiotic division happens in the gamete formation. So what happens? So the DNA content would always be half in the gametes and it would always be full and constant in a zygote. So that's how that constant number or constant content of DNA is maintained. So the only point you have to talk here is meiotic division or meiosis. So yes, that's it. Now let's move on to another topic in the chapter, which is variation. Yes, yes, hi, hello, hello, yes, hello, hi, hi, hi. Okay, okay, fine. So, variation. Through the questions that we have discussed, we have already said what variation is. Variation is the kind of process happening inside your body which would create a DNA that is similar to the parent but not identical to the parent. So that difference between me and you or me and my parent is the variation. Any difference between the cells, individual organisms or groups of organisms of any species is called variation. So it's basically the difference helps to adapt to new surrounding, and this is the same variation which would help me or a bacteria to survive in a harsh condition. And this variation can occur in my body or in a plant's body or in a microbe's body through mutations. Mutations are what? Mutations are nothing but they are sudden heritable changes. Sudden heritable changes. Heritable changes happening in your genetic composition, maybe in your DNA. You can say like that. So they are the sudden heritable. Heritable means it can go to your baby or you can get it from your parent. So sudden heritable changes which happen inside your DNA or in your gene composition is what you call as the mutation. So mutation is one of the reasons for variation to happen, and it can be sexual reproduction, the crossing over of two different chromosomes happening inside your body during the sexual reproduction can be another reason. So these are the different reasons by which variation can occur in our body. Okay, then some points to add on regarding variation is that variation during reproduction are the basis of evolution. So that's another major thing about variation. Variation happens through mutation or sexual reproduction. It is what helps us to adapt to a new environment. At the same time, this is the same variation which helps us, or this is the same variation which helps any species that are living to evolve. So that's the basis of evolution. So small variations, small differences in us gets accumulated, and that leads to evolution. So it's a basis of evolution, and variation that can thus be said as a raw material for evolution. So without variation, no evolution. Simple. Okay. So since we have said a lot about variation, let's see how these crossing over thing happens during sexual reproduction that can lead to variation. So during this sexual reproduction, what happens is, the suppose this is the paternal chromosome, and let's take another color for maternal chromosome. So suppose these are the two different sets of chromosome. One set, the blue set from your father side, and the purple set from your mother side. Okay. Suppose this is the condition. Okay. So such chromosome which are similar to each other, and that comes from the parents, from maternal and paternal side, which are similar to each other, are called the homologous chromosome. Homologous chromosome means they're so similar to each other that they would be carrying the similar kinds of alleles, similar kinds of characters in them. Okay. And during the sexual reproduction, or when the crossing over happens, what actually happens here is that they both come together in such a way that they are so close to each other that they almost link to each other, and that process is called synapses. They just glue to each other. That process is called synapses. Okay. And then after the synapses is done, they simply exchange their genetic material, and that process is called crossing over. That process is called crossing over. So it would be something like this. They would be exchanging their genetic material, and that particular process is called crossing over. This particular process called, sorry. Okay. So now they have come close to each other, and they're actually interchanging their genetic material, and that particular process is called crossing over. Crossing over. And once the crossing over is done, what we can see is a different set of chromosome, that we have never seen before. It would be something like this. It would be something like this. That would be the paternal chromosome after the crossing over, and the maternal chromosome after the crossing over would be like this. Yeah. So as you can see in the diagram, see, they have completely exchanged their genetic material. They have completely exchanged their genetic material in such a way that a new set of recombinant chromatids. Recombinant chromatids means they have already recombined, they have already exchanged. A new set of recombinant chromatids have formed here, and such a chromatid was never seen before. None of the maternal or the paternal set of chromosomes were in that combination. So a new set of combination has happened here, and that new combination is what you basically call as variation. Inside that recombinant chromatid part, where the exchange has happened, where the new chromatids are produced, there you would be seeing the variation, and that variation won't be there in the homologous chromosome. And this is the variation which is the raw material of any sort of evolution, and this is the variation which can make us adapted into a new environment too. I hope that concept is clear. Okay, fine. Moving on. Okay. Now let's get into the major content. So we have discussed regarding the blueprint, DNA, genes, chromosome, and variation, right? So that's almost the intro of the chapter. So yeah, that was just an intro. And from that intro part itself, we have got almost 2-3 questions, right? So it is possible to get questions from that particular region. Yes. Now let's discuss the major content of the chapter, that is reproduction. Reproduction is nothing but a biological process which leads to the formation of an offspring, right? And this offspring formation, or creating a new baby, creating a new offspring, can happen in two different methods according to biology. One is asexual reproduction and the other one is sexual reproduction. So reproduction can take place in two methods or two ways. One is called the sexual reproduction and the other one is called asexual reproduction. And I think everyone in here, whoever is attending this class, already know the difference between asexual and sexual reproduction. So I don't think I have to list it out and explain the differences, if I am not wrong. So sexual reproduction is a kind of reproduction where a male and female gamete would be there. They would be fusing with each other to produce that beautiful cell, a single cell called the zygote, out of which a future baby, right? A future baby would be coming. That is sexual reproduction. So if there are two parents involved and there are two gametes involved, then you can easily call it as a sexual reproduction, right? When you're going through the asexual reproduction, the difference is that there is no need of any gamete. You don't need a male or a female or anything. A single parent, it's a single parenting. You don't need papa and mama to be there. A single parent replicates itself or makes copy of itself to create a new offspring, to create a new baby. So that's the major difference between sexual and asexual. No gametes, gametes are involved. No fusion, fusion is involved. Like that. Okay. Now the question from that particular part is, what is reproduction? List its two types. How are the mode of reproduction different in unicellular and multicellular organisms? Yeah, it's a very simple question, very simple and basic question, I can say. It's a direct question too. What is reproduction? I think we have already discussed it, and list the two types. We just said it. So, reproduction is nothing but a biological process, right? Reproduction is nothing but a biological process, and it's a process by which new individuals or offsprings are produced from their parents or the parent through sexual or asexual means respectively. It is linked to the stability of population of a species. So reproduction is a kind of biological process by which a parent or parents makes the next generation, and there is definitely gametes or genes, chromosomes involved in it, and it can be sexual or asexual. That's a two mode of reproduction. Now the question is, how are the modes of reproduction different in unicellular and multicellular organisms? So again, that's a very simple kind of question. In unicellular organisms, so if I ask you to name some unicellular organism, what would be the first answer? Can I see it in the comments? If I ask you to name some unicellular organism, what would be the first thing that comes to your mind? Any unicellular organism in your mind? Excuse me. Yes. Okay. So when a unicellular organism, okay. So when you're talking about unicellular organism, the first thing coming to our mind would be an amoeba or a paramecium, right? Or it could be a bacteria. Okay, anyways, unicellular organisms like amoeba or paramecium or euglena, all those stuff, they are generally the ones who reproduce asexually. So most of the majority of unicellular organism doesn't need a complex process like sexual reproduction with two gametes, fusion, fertilization and all, they can simply replicate, they can simply make a copy of them by just doing a copy paste mechanism. Done. So they normally undergo asexual reproduction like fission, budding, fragmentation. There are different methods of asexual reproduction. And they have only one cell body and can multiply easily by simple cell division. So a simple normal mitotic division, just one parent splitting two daughters. That's simple as that. Okay. In the case of multicellular organisms, in multicellulars, they use both asexual and sexual mode of reproduction. So when you're going to a higher organism like a vertebrate or maybe a plant, the thing is they can do both sexual and asexual reproduction. Okay, then simple multicellular organisms usually follow the asexual method of reproduction, while the complex multicellular organism reproduce mainly by sexual reproduction. So you can simply elaborate that question like that. The question is basically what happens to multicellular and what happens to unicellular. So you can elaborate it in such a way that unicellulars normally undergo simple mitotic division and replicate because their body is very simple, right? And that is asexual reproduction. In multicellulars, the simple multicellular organisms can undergo asexual as well as sexual depending upon their need. While going through higher or complex multicellular organism, they normally depend upon sexual reproduction, obligatory. Okay, so asexual reproduction is where only one parent would be involved, and it occurs mostly in low group of organism, and there is no fusion of male and female gametes. While in the sexual reproduction, the production of new offspring by combining genetic material. I think we have already explained it. There could be a sperm and there could be an egg, and they would be forming a zygote. So there is that fusion of male and female gametes in the case of sexual reproduction, and during this particular fusion or fertilization, sexual recombination can happen, right? Okay, so here we need two parents of different sex and two gametes are needed there. So that's the major difference. Talking about asexual reproduction, where one parent is enough and no gametes are much needed, it can be classified into 6. According to the NCERT we are studying, according to your reader, the asexual reproduction methods can be classified into 6. The first one is fission. Second one is fragmentation. Third one is regeneration. Fourth one is budding. Fifth one is vegetative propagation. And the last one is spore formation. So these are the 6 methods of asexual reproduction which we have to go through. So right after these 6 topics are over, the major part of the chapter, which is asexual reproduction, would be done. Okay. Then the remaining is sexual reproduction, which we have to study in both plants as well as in human beings. So almost half of the chapter would be done if you are covering up these 6 topics. Okay. I hope you all are still here. So we'll go through each and every topic. Let's begin with the first question. So we'll discuss those 6 methods through some questions. I don't want to directly go to the content. I think you already clear about the theory. So let's discuss the question and let's see how the questions can come. And then we'll discuss the theory. So this is a question from the asexual reproduction part. It goes like this. Name the causative agent of the disease Kala Azar and its mode of asexual reproduction. So two things are asked there. You have to name the agent of Kala Azar and you have to name the mode of its asexual reproduction. So out of those 6 modes, you have to say which one is it. That's it. So two things have to be answered here. So I think you all already knew that the thing being talked here, the thing they're asking here is basically our leishmania. So Kala Azar, it's also called a leishmaniasis, right? It's also called a leishmaniasis. The causative agent of the disease Kala Azar is an organism called leishmania. It's an organism called Leishmania. This is the organism. This is the organism. This is a Leishmania. It's a flagellate organism. You can see the long flagella extending from its body that it used for this micro propulsion, we say. It uses that flagella to move. Okay. So the causative agent of this disease is called Leishmania, and the disease is also called Leishmaniasis. Okay. It reproduces asexually through binary fission. Yes, that's the first method of asexual reproduction, right? We already listed it. We listed 6 of them, and that's the first one, binary fission. So this is the answer to the question. So let's just go through that particular topic. Leishmania's, sorry, let's go through the topic fission or asexual reproduction, the first method. So fission is nothing but it is where a parent cell splits into 2 daughter cells. Simply, there would be a cell by some sort of mitotic division, they split into 2 different cells, and the new ones are called the new generation, the offsprings or the daughter cells. Okay. So, this kind of fission, this splitting up thing, simple mitotic division, it's a very basic form of reproduction. Almost all the simple unicellular organisms do reproduction through this method. It's very simple and very much less energy is needed for it, right? Okay, they don't even have to look for a mate. They don't have to go in search for a female or a male gamete. They don't have to swim. They don't have to wait. They can simply split, and yeah, next generation is here. So for unicellular organism, the fission is a very basic and most frequent method of reproduction. And there are different types of fission. The first one is what we just saw. It's called binary fission. And normally the animals like amoeba, the one we just explained, Leishmania, are the ones who undergo binary fission. Binary fission is nothing but, as the word suggests, see, it's binary fission. Bi means 2. So one cell splitting into 2, it's binary fission. Bi. Okay. So you can see that in amoeba. Even in amoeba also the same thing happens. You can see it here in the figure. Right. You can see it clearly. So this is one amoebic cell and it is elongating during replication. During its reproduction time, it's replicating. You can see the nucleus getting elongated. See, the nucleus have elongated in such a way that it elongated and is almost splitting. And by the end of the time of reproduction, they have actually become into 2 different cells with 2 different nuclei, and they splits. The final cutting happens and they split into 2 different organisms. So from one amoeba, 2 baby amoebas are actually formed just by splitting. That's it. So that's also a type of binary fission. And the one we have seen here in Leishmania, see, you should be very thorough with these figures. Okay, in this chapter the diagrams are really important. They can actually ask you to draw it. They can. So make sure you know all these diagrams by hearted. Okay, so there is this Leishmania and you can see a new flagella forming out of it. The nuclei inside it is splitting and it is actually becoming 2 nuclei. And yeah, see, from one nucleus it has separated and almost 2 nuclei are formed. Now once the nuclear division has happened, it's over. See, the body of the Leishmania is dividing now. The entire flagella comes out by that time and the body is now splitting. So by the end of the reproduction time, see, you can see 2 different Leishmania has actually formed. And at the end, what can happen is they split out of each other completely. So one will be one Leishmania, the other will be another Leishmania. So that's how simple that is. Okay, so that is also a binary fission. From one cell, 2 daughters. It's also a binary fission, 2. But then there is a special case of fission which is called the multiple fission. This term might not be very familiar, right? You might not be very familiar with that term. Like multiple fission is something a bit different from the binary fission. where you can see that particular example in an organism called Plasmodium. Plasmodium, have you ever heard about Plasmodium? Have you ever heard about Plasmodium? Yeah. So Plasmodium is nothing but a very small microorganism. It's a protozoan. It's a protozoan means very old animal. Okay. It's a protozoan which cause the disease malaria and all. It creates several other issues, but causing malaria is one of the major most issue from Plasmodium. So see, here you can see a Plasmodium. This is a single Plasmodium. And this Plasmodium, if you look inside that unicellular organism, inside that cellular membrane, you can see multiple nuclei. Multiple nuclei can be seen. And during this multiple fission, what happens is each of these nuclei would grow into one plasmodium. Each of the nuclei will develop a cell membrane around it, and each of these nuclei would become a single plasmodium. So once the maturation is over, this cell membrane would break off. The cell membrane would break off and multiple baby plasmodium comes out of it. Multiple baby plasmodium come out of it. So see, so by the time the cell breaks, not just one, but multiple would be coming out. So from one cell, multiple fission has happened, splitting has happened and created multiple organisms, multiple baby organism, offsprings. So such a division is called multiple fission and it can be seen in Plasmodium. So these examples under the heading fission, it's very important. So there are two types of fission. One is the binary fission, example amoeba, leishmania, and the other one is multiple fission, example plasmodium. Done. Okay. So yeah, here is an assertion and reason kind of question. You would be very familiar with it. So let's try to solve the question. Okay. Amoeba always produces two daughter amoebae, while Plasmodium divides into many daughter cells. That's the assertion. Now the reason is amoeba undergoes binary fission while Plasmodium undergoes multiple fission. I think from the theory that we have just learned, it's enough and more, right? It's enough and more. Okay. Okay. So what would it be? The assertion. It's definitely true. So check whether the statements are true. As I always say, this is how you have to come to the conclusion of an assertion and reason kind of question. Just check whether both the statements are true or false first. Then find whether the reason is an explanation or not. So first statement, it's true. The second statement, it's true. And since both the statements are talking about the same thing, this is the explanation of assertion. So yes, both A and R are true and R is the correct explanation of A. It's option A. Simple. Yes. Okay. Okay, fine. Now this is another question. Let's try to solve it. Name the process shown below and define it. Name the process shown below and define it. Have you ever seen such an organism? Have you ever seen such a figure anywhere in your textbook? So yes, definitely you would have seen this figure in your textbook. It's directly taken from NCRT only. It's an NCRT pic. Okay. And name the process. We have to define it. Name the type of cells present in the organism which exhibit this process. So what is this process they are talking here? What is this process they are talking here? I think you have an idea about it, right? The process which is being talked about is nothing but a method of asexual reproduction called regeneration. Okay, so they're talking about regeneration here, and I think you already have an idea. This is a creature, this is an animal called planaria, right? It's a worm, it's a flatworm. It's called planaria, and planaria is a flatworm, and it's a kind of creature which can split into different parts, and each of that part can regenerate into an adult. So it's a different kind of ability that it has, right? So it's the planaria has an ability that even if it is cut into two different or three different parts, each of that part can literally grow into a adult planaria. See, it takes few weeks, it takes time, but still it can actually grow back. It never dies. It is not dying, it is growing back into a new organism. So the process shown here is called regeneration. This particular type of asexual reproduction is called regeneration, and this is another method of asexual reproduction out of the 6. Name the type of cells present in the organism which exhibit the process. Okay, so they have not only asked us to talk what the process is, they have asked to explain the process. So we have to explain what regeneration is. So the process is regeneration, and regeneration is the ability of an organism to give rise to a new individual from any broken or injured body part. So even if an injury has happened, or even if you cut the planaria into two, it can actually grow back into a completely grown adult. And it is considered to be a form of asexual reproduction in lower group of animals. So regeneration normally, majority it happens in the lower group of animals. At the same time, if you take a liver cell in humans, in mammals, the liver cell has the capacity to regenerate, right? So regeneration capacity is there for some of our stem cells. So not only lower group organism, even the higher group organisms has shown the ability to regenerate. Anyways, so this is what you have to explain about regeneration. And the second question is, what is the kind of cell which is present in that particular region of planaria which helps it to regenerate? So what is that particular type of cell? So that's the kind of cell. In the NCERT, they have talked about it in a word using specialized cells. They have used the word specialized cell. So inside the animal planaria, there is particular regions with specialized cells, and these specialized cells are what we basically call as the neoblast, or they are basically some stem cells, you can say. So stem cells are nothing but they are the cells which have self replicating self replicating or rejuvenating capacity. They can rejuvenate, they can always be new, they keep replicating in such a way that they can generate new cells from them only. So they have self-replicating capacity and they are rejuvenating. They're never going old. They always rejuvenates. So such stem cells, which are called a specialized cell in your reader, it's actually called the neoblast. Such specialized cells are called the neoblast. And the regions of the planaria where such neoblastic cells are present, you can actually see the regeneration capacity. Understood? So that regeneration capacity goes to the part of the worm where neoblast or the specialized cells are present. Okay. And yeah, what happens when a mature Spirogyra filament attains considerable length? Okay, again, it's a similar kind of question from the same topic. It's again asexual reproduction. So what happens when a mature Spirogyra filament attains a considerable length, or on maturation sporangia burst? So they're asking two different types of asexual reproduction in the same question. So the first question is regarding Spirogyra. So Spirogyra is nothing but it's an algae, right? It's a very low group of plant. When a Spirogyra filament attains a considerable length, it breaks into small pieces called fragments. It's called fragments. It's not a regeneration, but when it's an adult, the plant or that algae itself breaks into small pieces, and from those small pieces, each of these fragments grows into new individual. It's different from regeneration in such a way that there is no specialized cells acting here. There is no neoblast here. Okay, it might look similar, the regeneration and fragmentation, but here there is no specialized cells, and this mode of reproduction is called fragmentation. And okay, suppose this is a Spirogyra cell. Suppose this is the long filament. So when you look into the spirogyra under a microscope, it looks very elongated, multicellular organism. Okay, with a very greenish bluish color. So each of the cells might look like this. And when the spirogyra splits into small parts, each of these part, suppose this is one part of the spirogyra and this is another part of the spirogyra. Each of this part of the spirogyral filament can actually grow into a, an elongated, mature adult, just like the parent. So each of them has the capacity to grow into an adult filament. I hope it makes sense. So this is called a spirogyral fragmentation. So each of these parts, which are the broken out pieces from the parent, is what we call as the fragments. So these are the pieces. Basically, it means pieces. So these are called the fragments, and each fragment has the capacity to grow back into a new spirogyral filament. Okay, so such a method of reproduction is what you call as fragmentation, and fragmentation example is spirogyra. Okay, and yeah, there is one more question. I think with that we'll wind up the session. This is the question. On maturation of the sporangia, it bursts, right? On maturation, the sporangia bursts. So they're asking what is happening here. So by the term sporangia, you should have an idea what they're talking about in the question, right? So they're talking about nothing else. When a sporangium bursts, a large number of spores are released into the air. When the spores land on food or soil or any moisture environment, under favorable condition, they germinate into new individuals. Okay, so one second. Where is it? Okay, so this is the sporangia they are talking about. This is the same diagram taken from your reader. See, this is a fungus called the rhizopus. This is a fungus called rhizopus. So this is also called a bread mold. You might have heard about it. This is an organism called rhizopus or bread mold. It's a fungus that grows on the on an expired bread piece or a cake if you keep it open or if you kept it in a moist condition, right? So this fungus, it looks like this. It has a long stalk on which bulb like structures can be seen. Okay, it has a long stalk on which the bulb-like structures can be seen. So these bulb-like structures are called the sporangium. They are called the sporangium. And inside the sporangium, see here in the figure you can see, inside the sporangium there are thousands of small, minute seed-like structure. They are microscopic, and that is called the spore. So there is sporangium inside which spores can be seen, and then there is this long stalk, right? The stalk, and yeah, they have this hyphae. Hyphae is nothing but the root-like structures, you can say. So this hyphae or the root-like structures helps the fungus to, what do you call, to stay attached to the substratum and then keep multiplicating. Okay, so yeah, this is rhizopus. Okay, one second. Yeah. Hyphae are present in the rhizopus, tiny blob on a stick structure. These blobs are the sporangia, sporangium in singular, which contain the spores. Spores can be developed into a rhizopus individual. So when this spore falls on a particular moist environment, there should be enough water and conditions. And when it falls on a moist condition, uh, they can grow into rhizopus individuals. And also these spores are covered with thick walls. So if you take look into a spore, it has very thick coating around it, which makes it very stable. It's almost thermostable, pH stable. It can withstand almost all kind of harsh environment. So it can stay alive for a long time. and these walls protect the spore until they come in contact with another moist surface. So even if they fall down, they can stay there like that for few days, maybe few months, until they get a new, what do you call, a new space to grow. Understood? Okay. So okay, sorry. So when sporangium bursts, a large number of spores are released into air, and the spores, when it falls on particular food or soil with enough moisture content there, they can again grow up into a new rhizobus, a new fungus. Understood? So this is again another method of asexual reproduction, which is called spore formation. This is a method of asexual reproduction called the spore formation, and the one we have talked here about the spirogyra, the splitting of the filament, that one is called the fragmentation. This is called the fragmentation. Fragmentation. Yes. So fragmentation, spore formation, fission, multiple fission, binary fission. I think almost all of those six are discussed, but still we have a long way to go. So with this particular question, I'll wind up this session and I'll be back with the next part. So yes, I hope you will keep all this content in your mind, and I hope we'll be seeing each other for the next session. So thank you so much. Thank you. Thank you so much for staying with me. Thank you so much for the patience. I hope to see you all again back in the next session. So yes, that's it. That's bye from me, Alkara Jimon, your biology tutor from GoSco. Take care and good night.